Somewhere in the rainforests of Central America, South America, and Southeast Asia, there is an animal that looks like nature could not decide what to build. It has the snout of a tiny elephant, the body of a pig, and the feet of a rhinoceros. Most people in the United States have never seen one outside of a zoo, and many have never heard of it at all. But scientists who have spent years studying its genetic code say the tapir is hiding one of the strangest and most overlooked stories in the entire animal kingdom. What they found inside its DNA does not just explain what a tapir is, it rewrites what we thought we knew about horses, rhinos, and the deep history of mammals on Earth. Let's start with the basic mystery. A tapir looks like a mashup of at least 3 unrelated animals. Long before genetic testing existed, naturalists guessed that tapirs were some strange cousin of pigs because of their stocky bodies and short legs. Others assumed they were related to elephants because of that flexible trunk-like nose. But when researchers finally sequenced tapir DNA and compared it against other mammals, the results shocked everyone. Tapirs are not related to pigs at all. They are not related to elephants either. Genetically, their closest living relatives are horses and rhinoceroses. All 3 of these animals belong to a group called Perissodactyla, which scientists often call the odd-toed ungulates. That means their body weight rests on an uneven number of toes, and their digestive systems are built the same unusual way, fermenting plant material in an enlarged section of the gut rather than in a multi-chambered stomach like a cow. On the outside, a tapir looks nothing like a horse or a rhino. But underneath, buried in millions of years of genetic code, the 3 animals are telling the same story. They came from a shared ancestor. Here is where it gets even stranger. Researchers digging in an old coal mine in India uncovered fossilized bones from a creature called Cambatherium, which lived around 54.5 million years ago. When scientists studied its skeleton and compared it to genetic and anatomical data from modern perissodactyls, they realized this ancient creature was not quite a horse, not quite a rhino, and not quite a tapir. It was something like a cousin standing just outside the family, incredibly close to the actual point where horses, rhinos, and tapirs split apart from one shared lineage. At the time this animal was alive, India was not attached to Asia. It was a massive island, still drifting across the ocean, slowly making its way toward a collision that would eventually connect it to the Asian continent. Somewhere on that drifting island, one of the strangest evolutionary origin stories in mammal history may have begun. Later, genetic research pushed the estimate even further, calculating that the shared ancestor of horses, tapirs, and rhinos existed somewhere around 65 million years ago. If that number is accurate, it means the lineage that eventually produced tapirs was alive around the same time period as the mass extinction event that wiped out the non-avian dinosaurs. While dinosaurs were disappearing from the planet, an early ancestor related to the modern tapir may have already been quietly establishing itself. For a long time, tapirs earned a nickname among biologists. They were called living fossils because their basic body shape has barely changed for millions of years. Fossilized tapir-like creatures found from tens of millions of years ago already show that unmistakable flexible snout and stocky frame. It looks almost identical to a tapir walking through a forest today. But here's the twist that most people never hear about. While their outward appearance stayed remarkably stable, their DNA did not sit still at all. Genetic research shows that tapirs have been splitting, diversifying, and drifting apart at the molecular level for an extremely long time, even while their bodies looked frozen in time on the outside. Right now, there are 4 widely recognized species of tapir living on Earth. There is the Malayan tapir, also called the Asian tapir, easily recognized by the bold black and white pattern across its body. There is Baird's tapir, found in Central America. There is the lowland tapir, sometimes called the South American or Brazilian tapir. And there is the mountain tapir, also known as the woolly tapir, which lives at high elevation in the Andes and is covered in a thicker coat adapted to cold air. Genetic studies estimate that the Asian lineage split away from the American lineages somewhere between 20 and 30 million years ago. That is an enormous stretch of time, roughly comparable to the evolutionary distance between some primate lineages that look far more different from each other than any two tapir species do. Then in 2013, a team of researchers announced something that stirred up serious controversy in the scientific community. They claim to have identified a brand new species of tapir living in the Amazon, which they named the Cabomani tapir. If confirmed, it would have been the first new tapir species described by science in roughly 100 years, and one of the largest new land mammal discoveries in modern history. Naturally, this made headlines, but not long after, more detailed genetic analysis was carried out, and the results told a different story. When researchers looked closer at the DNA, the supposed new species did not hold up as genetically distinct. Current evidence suggests the Cabomani tapirs is not actually a separate species at all, but rather a regional variation within the lowland South American tapir. It is a perfect example of how DNA can either confirm or completely dismantle a discovery that seemed obvious just by looking at an animal's body. Now here is one of the most fascinating and least talked about discoveries in tapir genetics. Scientists compared the chromosomes of the different tapir species and found something unusual. The Malayan tapir, the species that is geographically isolated in Southeast Asia, has a noticeably different chromosome count compared to the three American tapir species. It also shares fewer chromosomal similarities with its American relatives than researchers expected. In other words, the geographic separation between Asia and the Americas is reflected directly inside the chromosomes themselves. But the story gets even more unexpected. You might assume that geographic closeness always predicts genetic closeness, meaning animals living near each other should be more genetically similar than animals separated by an ocean. Tapir DNA proves that assumption wrong. Researchers found that the Malayan tapir from Asia, Baird's tapir from Central America, and the lowland tapir from South America all share nearly identical X chromosomes despite being separated by enormous distances and millions of years of separate evolution. Meanwhile, the mountain tapir, which lives much closer geographically to some of its American cousins, has a distinctly different chromosome structure, with an added or missing segment of genetic material that sets it apart. This means physical distance on a map does not always match distance on the genetic level. Sometimes the animal living on the opposite side of the planet is genetically closer to you than the one living in the same mountain range. Scientists also trace the timeline of when the two South American tapir species diverged from one another, and the DNA evidence lines up almost perfectly with a major geological event. Millions of years ago, North and South America were not connected. They were separate land masses divided by open ocean. Then the Isthmus of Panama slowly formed, creating a land bridge between the two continents. This triggered a massive wave of migration known to scientists as the Great American Biotic Interchange, where animals from North America moved south and animals from South America moved north, permanently reshaping the wildlife on both continents. Genetic research shows that the split between the two South American tapir lineages happened at almost exactly the same time as this land bridge formed, roughly 3 million years ago. In other words, the geological history of an entire continent is written directly into tapir DNA. Now let's talk about something most people watching this have absolutely never heard about, because it barely makes headlines outside of scientific journals. Conservation geneticists studying tapir populations have discovered a serious and quietly alarming problem. Every single one of the 4 tapir species is currently struggling with dangerously low genetic diversity. This is not a minor technical detail. It is one of the biggest threats facing tapirs today, arguably even more dangerous in the long run than habitat loss alone. Here is why genetic diversity actually matters. A wild population with plenty of genetic variety can adapt to new diseases, changing environments, and unexpected threats because different individuals carry different genetic tools for survival. But when a population shrinks and becomes isolated, animals are forced to breed with close relatives simply because there are not enough unrelated individuals left. This is called inbreeding, and it slowly narrows the genetic toolkit available to a species, making every future generation more vulnerable. Researchers studying the Malayan tapir recently carried out the very first genetic investigation of something called the MHC gene family in this species. MHC stands for Major Histocompatibility Complex, and in simple terms, this is the part of an animal's DNA responsible for recognizing dangerous invaders like viruses and bacteria, then triggering the immune system to fight back. It is essentially the genetic blueprint for disease resistance. When scientists analyzed the Malayan tapir's MHC genes for the first time ever, they found alarmingly low diversity. With only an estimated 2,000 to 2,500 Malayan tapirs left in the wild, low immune diversity means a single new disease could spread through the population with far less genetic resistance to stop it, potentially threatening the entire species at once. Similar warning signs have shown up in Baird's tapir, the Central American species. Multiple genetic studies using DNA extracted from blood, hair, and even feces have shown that wild and captive populations face a rising risk of losing genetic variability due to inbreeding. Some researchers have pointed out something particularly sobering about tapirs. Because they do not have many closely related living species, if a tapir lineage goes extinct, it does not just mean losing one type of animal. It means losing an entire branch of the evolutionary tree that cannot be replaced or recovered from a closely related cousin species, the way it sometimes could with more genetically diverse animal groups. Even more surprising, full genetic mapping of tapirs has happened remarkably recently and shockingly late compared to many other large mammals. The complete mitochondrial genome of the Asian tapir was not fully sequenced until 2014. It turned out to be a sequence just over 16,700 genetic letters long. The complete mitochondrial genome of Baird's tapir, an endangered species native to Central America, was not fully sequenced and published until 2022. Researchers described it as the very first genomic resource ever created for that species. Think about that. In an era when human genomes, dog genomes, and even genomes for extinct woolly mammoths have been mapped out in extraordinary detail. One of the largest land mammals in Central America only received its first complete mitochondrial genetic map within the last few years. Tapirs have essentially been hiding in plain sight, understudied, and genetically mysterious right up until now. There is another discovery buried in tapir DNA that almost nobody outside of specialized research circles has ever heard about. Scientists studying wild lowland tapirs in Brazil collected blood samples and ran genetic tests, searching for a group of bacteria called hemoplasmas, which are blood parasites capable of infecting red blood cells. The results were startling. Out of the wild tapirs tested, roughly 1/3 came back positive for these bacterial infections. Even more interesting, the genetic sequencing revealed strains that appeared to be entirely new, not previously documented in scientific literature. In other words, hidden inside the bloodstream of these massive rainforest herbivores, researchers may have uncovered previously unknown bacterial species simply by sequencing DNA extracted from routine blood samples. And it does not stop at bacteria. Researchers have also used a cutting-edge method called DNA metabarcoding to study what tapirs actually eat in the wild. Instead of following an animal around with a notebook, trying to observe every bite it takes, scientists can now extract and sequence tiny fragments of plant DNA preserved inside an animal's droppings. When this method was applied to tapir feces collected in a protected rainforest region of French Guiana, the genetic data confirmed information researchers already suspected about the tapir's diet, but it also revealed something new. The sequencing identified plant families and even entire plant genera never before recorded as part of the tapir's known diet. This matters far beyond just satisfying curiosity about what a tapir likes to snack on. Tapirs are considered one of the most important seed dispersers in the rainforest ecosystems where they live. As they wander for miles eating fruit and vegetation, seeds pass through their digestive system and get deposited across the forest floor, often far from the original parent plant. Some researchers refer to tapirs as gardeners of the forest for exactly this reason. If their diet is broader and more complex than previously understood, that means their overall ecological role in maintaining rainforest biodiversity may be even bigger than scientists originally gave them credit for. Even the tapir's most famous feature, that strange flexible nose, holds a piece of unexpected biological information. Unlike an elephant's trunk, which contains a complex internal skeleton supported by cartilage and muscle in a very specific structural arrangement, a tapir's proboscis is built almost entirely from soft tissue without significant bony internal support. Researchers studying the evolution of this feature have noted that the length of the nose varies noticeably between species. The Malayan tapir has the longest snout of any tapir species, while the lowland South American tapir tends to have the shortest. Because this structure is made almost entirely of soft tissue, it does not preserve well in the fossil record, which is part of why genetic and anatomical research has become so essential in understanding exactly how and when this feature evolved across different tapir lineages. So what does all of this add up to? A shy, quiet, often overlooked rainforest animal that most people in the United States could not identify in a lineup has turned out to be one of the most genetically revealing creatures on the planet. Its DNA connects directly back to an ancient island that used to drift freely before colliding with Asia. Its chromosomes reveal that geography does not always predict genetic relationships the way we assume. Its genetic divergence timeline lines up with the exact geological moment when North and South America became connected by land. Its immune system genes are dangerously low in diversity, quietly putting entire species at risk in ways that rarely make international news. Its bloodstream may be hiding previously undiscovered bacteria, and its droppings, of all things, are rewriting what scientists thought they understood about its diet and its role in keeping rainforest ecosystems alive. The tapir looks like a strange leftover creature that evolution forgot to finish designing. But its DNA tells a very different story, one of ancient survival, hidden diversity, deep evolutionary connections to horses and rhinos, and a present-day conservation crisis that almost nobody is talking about. The next time you see a tapir, whether in a documentary, a zoo enclosure, or a photograph online, remember that this strange-looking animal is quietly carrying one of the most surprising genetic histories in the entire mammal kingdom, written in code millions of years in the making, and scientists are still only beginning to fully read it.